Well, thank you all for hanging in and for the good back and forth of the sessions this morning. Really do appreciate it, and hope that you all appreciate, as I appreciate, the interaction and the importance of what we're doing here, but more importantly still, what comes after this. And we'll have a chance to follow up with each of you on your impressions of the workshop and how we can do better going forward, but more importantly, how we kind of narrow down what the focuses need to be for our next convenings and help us with strategy. So when we first framed the upcoming session, we invited Dr. Chambers to join us. It really was based on our belief in that there is clear data suggesting that there are functional deficits in early stage disease, and whether it's in NPDR or even antecedent to evidence of any vascular change. And so for us who want to prevent progression to vision-threatening diabetic retinal disease, there is a clear unmet need to examine what interventions are possible in those earlier stages that can be distributed to a wider population and prevent progression. And in that case, if you're asking the question about what can we do to treat earlier stage disease, you have to understand how you classify the disease, how you identify patients who are at risk, and what you ultimately are able to measure that will help in a process of therapeutics development that can show not just a stage of disease, but also a treatment effect. So these are big challenges. And so when we framed this session, I created a placeholder title for this, which was Regulatory Pathways in Neurodegenerative Ocular Disease. The reason I framed it that way was really all about, can we learn from other ocular diseases the sorts of things that we need to start to measure and codify as potential useful endpoints to guide clinical care, diagnosis, clinical care, but also the development of new drugs and their approval. So I'm going to invite Dr. Chambers to come and provide perspective on endpoints in ocular disease related to diabetes, but also where he's able to give us some insights or share with us if there are insights we can gain from other ocular diseases that will help us in our process of divining the endpoints that are required to accelerate new therapeutics development and better diagnosis and treatment. So Dr. Chambers, thank you.
Thank you very much. So this is my standard disclaimer. These are my views, not necessarily those of the Food and Drug Administration's, and I am prohibited by law from having any financial interests either in any manufacturers or any competitors, so I don't. The basis for approval decisions all comes from the Food, Drug, and Cosmetic Act. And that is very explicit and talks about safety and efficacy being determined by adequate and well-controlled investigations. The bottom line to that is everything else doesn't count. And as much as any of the non-clinical information leads you to some particular area or some assay suggests or some mechanism of action suggests, if it's not demonstrated in adequate and well-controlled trials, it doesn't count for approval. But it only has to work for what it says it's going to work for. It doesn't have to be a panacea for everything. As long as it works and the people that the product is labeled and defined as working in, that's good enough. But the law also specifically excludes us from using isolated case reports, random experience, reports lacking details, or any uncontrolled trials. And you see that's frequently the initial basis, and there's been some discussion earlier this morning about what leads you to getting there, but it's not what the Food and Drug Administration uses for approval. What are adequate and well-controlled trials? Well, it's a study design that permits a valid comparison. So you have comparator arms doing similar schedules where the patients can't tell which group they're in. Multiple different things in the trial to try and minimize bias. on the part of subjects, observers, analysts. People should not know until the trial is over which group they were in, so that they don't give a false sense of efficacy or don't report aspects of safety. And the method of assessment has to be well-defined and reliable. And that's where this consistency comes in, that you see people frequently say, well, it's not quite good enough, or what does it need to be validated? We're looking for, is it well-defined and is it reliable? For diabetic ocular conditions, currently we separate two different indications. We have treatment of diabetic macular edema and we have treatment of diabetic retinopathy. You can argue that there's overlap between the two, there certainly is, but we think there are significant differences, and products may do one, may do both, or may do the other, or may do both. For diabetic macular edema, we accept improvement in visual acuity by at least 0.3 logmar. We don't care if it's in high contrast, we don't care if it's low contrast, or something in between. That works in both directions. Not only can you show improvement, you can show prevention of loss. You can do it either as a mean change, or you can do it as the percentage of subjects that show improvement. Any of these things, we think, show a clear improvement in the diabetic macular edema. And why is visual acuity important? I think I mentioned earlier today. You cannot drive in the majority of countries, majority of states in the United States, unless you have a certain visual acuity. And until we change all the laws in all the different states, it's critically important for the independence of most people in the United States. Yes, there are cities where people walk around and don't need a driver's license, but large areas of the country, you need a driver's license to be able to be independent. And so it becomes critically important. Is it the only thing that's important? No, of course not. But it is critically important. We do accept other functions of, other measures of visual function. And we usually describe it as visual function, not as just visual acuity. Yes, visual acuity is one measure of visual function, but there are plenty of others. Visual field is equally important. We've gone and defined an amount that is of change in visual field that's clinically important. That being greater than, picking a predefined area, having that area change by 7 decibels, and the area has to be a definable area. It's not a single point, and we've arbitrarily said at least 5 points. Microperimetry, same thing. You're measuring essentially the same thing. These are things we've defined. No questions asked. That's a clinically significant change. Does that mean that that's the only amount that's clinically significant? No, not necessarily. We may take something that's less than that, but only after we see the results of the clinical trial. This is one side of a coin. That's this benefit to risk. And if we don't know what the risk is, it's very difficult to make that comparison. While we've said this is unquestionably efficacy, anything less than that we want to balance with what the risks are. Diabetic retinopathy, we've also identified particular endpoints. The ETDRS scale was developed, and it's not the scale that was important, it was its ability to predict what was going to happen 10 to 15 years later. That's what made this scale important. Yes, it's old. Yes, there are pictures that don't go out to the periphery, but it was predictive of what happened as far as what the ETDRS called significant visual loss, which was 6 lines of change, but 10 years later. A minimal change was what was considered 3 lines. But so, kind of the same thing, we will take a change on the ETDRS, either measuring both eyes, and the scale was originally developed to be for both eyes. So a 3-level change in both eyes, or for those products that are administered only to a single eye, a 2-letter change. And again, both directions, either improving or preventing loss. There are other anatomic features that we take as far as endpoints. Preventing loss of photoreceptors. We have 2 products that are currently approved based on loss of photoreceptors. Preventing retinal detachments. You heard Ryan talk earlier about the difficulty of having a retinal detachment and the potential downside consequences. Preventing retinal detachments is a good thing in any situation, so we take that as an endpoint. We'll take prevention of nerve fiber loss as being a legitimate clinical endpoint. The question becomes, do we really need anything else? And I would argue our problem is not that we don't have endpoints. I think we have endpoints that can be used. The biggest one we'd like to see is showing the ability to do some activity of daily living that the person couldn't previously do. Trying to do things that benefit the patient is where we're trying to focus medications. Changing an anatomic feature that the patient's never going to notice, maybe the clinician will or will not notice, maybe it will change at various points in time, is not what our goal is. Our goal is doing treatments that benefit patients. I think the lack of our current neurological endpoints in diabetes is the fact that we haven't had products that can go and alter it. It's not the fact that we don't have the ability to measure it or that we haven't defined a particular change as being clinically significant. If you have a product that's going to change neurological function, we'll notice it in what we currently do. And I thank you very much for your attention.
So with that, maybe I turn to Dr. Aiello, and we can ask the question of how do we establish what the threshold is for clinically meaningful change as it relates to the neurodegeneration that's associated with diabetic retinopathy? And is there a threshold that will allow us to satisfy what Dr. Chambers is discussing as it relates to ADL? I'll reflect on personal experience for a moment, and it's similar to what Ryan had talked about. Marianne, progression of her disease, was troubled by things like limited vision in low light, reduced peripheral vision, and other attributes that really weren't connected to her best corrected central vision. She still had, she had preserved central vision, but she had difficulties to the point where she was no longer able to safely walk across a room, and yet she still had preserved central vision. So the question is, what would be a threshold effect that we could measure that might give us insight as to again, the sort of thing that might not be reported by the patient, but nonetheless is a definable, measurable functional loss?
Well, I think that it's an important sort of aspirational question, but I think it's very difficult or impossible to answer right now. I think the key aspect here is you are trying to look for things that relate to something that impacts the patient, as Dr. Chambers said. So obviously vision does, and the other endpoints that he pointed out. But there are other things that can impact the patient's life that we need to be able to show that there's going to be a benefit there eventually from that standpoint. Then you're going to need to know how well whatever you're looking at relates to that. What is your variability and so forth. We have a great example now, something that you can look at and tell major change. Central retinal thickness with an anti-VEGF, it goes from 400 microns to perfectly normally looking, and that's great. But because of the variability of its association with visual acuity, even though in aggregate it's well associated, the variability is so much that at 300, 350 microns, you can go from 20/20 to 2200. So you can't use that as an endpoint, and yet it's very informative to what we do. If you're going to go for the endpoints, you're going to need something that's going to affect either the accepted endpoints or ones that can be shown to work with the patient or have a predominance of the evidence, because FDA will look at your whole trial when it comes in. I think it's particularly important also because we're starting about only in this area, but so many new approaches to what we can look at, so many pathways we can look at in detail now that we're able to look at processes presumably upstream of even the late-stage diseases. And so these may be earlier to look at or better to look at or helps differentiate, and we're going to need to be able to show how well do they relate to accepted endpoints, and then can they be eventually be used as a surrogate for that with the proof that's there. And then even if you reach that, there'll be the next step. Can you go back beyond that? But when you go beyond that, you're probably going to not only have to show it's good for your new surrogate or earlier endpoint, but that relation to eventual visual loss is still there. But once those studies need to be done so rigorously that they're well known, they're well accepted, just like the FDA accepted endpoints, so that once that's shown, then we can use these going forward as the basis for other things.
Yeah. Thanks, Lloyd. Go ahead, Wiley.
But I want to take you back to, if you can't see in the periphery, that shows up on a visual field.
Yes.
So that will meet what our current expectations are already.
Thank you. Fair enough. Thank you. Ramin, Dr. Tadayoni, Lloyd's comments lead well to a conversation about what you're doing with EviRed. If you describe to the audience what EviRed is and what your expectations are for that longitudinal trial.
So the EviRed project is a French government-funded project in which we have included around 3,000 patients that we follow up for a few years. So the idea is basically to propose a new classification to estimate the risk of a patient of losing vision or going to complication. Because the problem with the ETDRS classification is that when you look to the literature, so it says that if you are level 55, you have 50% of risk of having a PDR in a year. All the new studies, when you look to the arm which is not treated, it's around 10% or 15%. So it's no more working. It was developed in a time when the diabetes control was different. And now that the diabetic patients are controlled differently, this classification is not working. It doesn't allow to select the patients to which we have to propose a treatment. So we collected a lot of images, all the data available for these 3,000 patients. We follow them from 1 to 4 years, depending on when they are included. And then we will see if we are able by AI or any other algorithm. So we have an AI program, but we are open to any other algorithm or classification that allows, again, to pick people who are at risk have 50% or more of having a complication that is clearly associated with loss of vision.
Yeah. Thank you, Ramin. Dr. Chambers, I will ask you again, you talked about two indications for intervention, DME and diabetic retinopathy. When we coined, if you will, with help from our working groups, the term diabetic retinal disease, it's as an umbrella term to include diabetic retinopathy, DME, but also what we call diabetic retinal neurodegeneration. Is there a value in for us to start to define a whole new category of diabetic retinal neurodegeneration as a means through which we can organize the community to start to address these issues?
So as I started to say, I think the problem is having come up with compounds that go and do it.
Yeah.
Not that we can't measure. We can already measure those functions. They're likely to show up in nerve fiber layer loss. They're likely to show up in visual field changes. I'm not going to begin to argue that visual acuity is the best way to go and measure it. It's not. But there are plenty of other measures we do within the eye. I mean, we have an advantage that a lot of other fields don't have. We get to actually look at what's happening at our tissue level. We have a direct measure of the function of the organ we're trying to preserve. You see a whole lot of surrogates get used in a lot of different areas because they don't have that ability to do the things we can do. We've been fortunate, we've taken advantage of the fact that we can get direct endpoints and not have to go to surrogates.
Yeah, appreciate it. Dr. Ehrlich, you've been, both at small biotech, you now lead a new one called Ollin, but you also come out of the big pharma and helped develop Lucentis. And give us a sense, from your perspective on how to address this issue of how do we identify targets for early intervention, preventive strategy that might be useful in folks who don't have definable diseases that we currently define it from the standpoint of vascular disease.
Yeah. So complicated question.
Yeah.
I guess a few a few points on it. The first is, how can you design a study like an interventional trial, right? And to try to address, whatever the endpoint is. And for instance, I think that's why, like some of the work that Ramin and others are describing of updating, or at least our current understanding of disease progression rates, be it for the DRSS scale, be it for, functional preservation or just change in vision, contrast sensitivity, any of these features in the modern era with modern diabetic systemic therapies and the current patient populations and the diversity of patient populations. Understanding the natural progression rate is really important for actually being able to design any interventional trial. And that's actually an area that a lot of the current knowledge is lacking, especially for new endpoints, right? So it's a little bit of a chicken and an egg problem, but if you're developing a new endpoint, this is where I think a lot of the work that the group described this morning is going to be important. How long does it take for that for a clinically meaningful change to occur? Without treatment, let alone with some new treatment. And if you're at a big pharma company, right? The portfolio, your projects have to sort of measure up in ability to measure proof of concept in the same period of time as, say, an oncology product or a blood sugar control product or a medicine for hyperlipidemia or any of those other things. If you're at a small biotech, at least my experience, right, is that investors in small biotechs, there's certain risks that they want to take, right? They want to invest in a new mode of delivering a therapy to the patient or a really novel, impactful biology, and they try to de-risk it through genetics or through animal models or things. What they don't really want to take a lot of risk on in early biotechs is new endpoint development, right? It takes a long time. The outcomes aren't really that clear when you're starting. So I think it's a challenge for diabetic eye disease in a small biotech. I think most people would really only want to work in areas that have already been well defined, how you would design and run a study. And there's not a lot of capability within small companies of investing in endpoints development because it's not really seen as the remit from the investors.
Yeah, obviously one of the reasons why we're doing what we're doing is to try and bring the community together to start to solve some of those issues. Chris, from the perspective of, maybe you talk a little bit about patient burden and how we utilize an assessment of patient burden in our decision-making process about development of therapeutics.
Well, so as a type 1 diabetic of 35 years now, for at least the last 15 years, I've had roughly every 6 months or so assessments. And wide range of assessments, from visual field to OCT and whatnot. And while I think those have been great to track the development of disease, what I think they lack is really a predictive potential and a capacity to tell me they're prescriptive. They can tell me that I need to go on therapy, but they cannot tell me I should start intervening or provide any real guidance for what I can be doing now to prevent the development of disease down the road. So just in that regard, I think developing biomarkers that are obviously have meaningful endpoints, but are predictive of disease course, disease onset, and really guiding intervention, in my opinion, would be extraordinarily helpful and extraordinarily valuable. I guess another component is thinking about the likelihood of me having to undergo these therapies and these interventions down the road. What we currently have, particularly for kind of end-stage disease, seems to be very effective, but not without significant burden. Not just of cost, anti-VEGF injections are not cheap, but also side effects, risk to my kidneys, which is another side effect of type 1 diabetes, is risk to the kidneys. And so is the therapy that is helping my vision or prevent loss of my vision impacting another component of my life negatively, and not to mention just the day-to-day burden of dealing with it all. I have to deal with managing my diabetes, going to the endocrinologist, all of that on top of just life, and do I want to take on something else, right?
Yeah.
that can prevent me from just living my life in an unburdened fashion. And so I guess I would really implore anyone who is thinking about intervention and developing interventions to really consider just the day-to-day impact as well as the burden that you're putting on someone that already has a lot of burden of disease management. Retinal disease is a disease of the eye, or at least diabetic retinal disease is a disease of the eye, but it's not because of the eye. It's because of the underlying disease. It's the diabetes that is driving it that has to be managed otherwise, and the eye is a primary loss of function, but not necessarily the primary one.
Yeah, thanks, Chris. So picking up on your comment about predictive ability, I'm going to take a risk of breaking through the wall here and ask Dr. Chang if he's willing to comment on the Bridge to AI project. You mentioned it earlier in your remarks, and how we can possibly add on to the work that's being done in that perspective, observational study, so that we can actually start to gain the sorts of insights that we're looking for our specific purposes.
Yeah, Robert, thanks for asking. I think that there's a lot of potential ways to answer. Robert, thank you for asking. I think there are a lot of potential ways to answer your question, and maybe I'll stick to one of them just for the sake of simplicity. And that'll be in the theme of data sharing and data harmonization. That I think that my observation is that we're really good at sharing data among ourselves and our friends/collaborators. And what I see often is cases where one group of friends develops one method to work together and another group of friends develops a parallel method, and they're just not the same. And I just think it would be a huge win for the community to be able to try to work on projects where we contribute to the same ecosystem. And I love having met a few people, Ramin, others who are building cohorts, similar to Bridge to AI. And I think it would be great to do it as if we're kind of all on the same team. So thanks for asking about that.
Yeah, no, thank you. I'll offer an opportunity for others to kind of contribute, to make suggestions. This is a little bit of a Gordian knot. I mean, we all recognize that there's early disease. We all recognize dysfunction in patients who have early stage disease, but we haven't really defined well what's clinically meaningful change. And as I said, on certain occasions, people won't report change, but they'll have change in their lives and their behaviors. Willie, go ahead.
I want to separate two different things. One is the endpoint, and the other is the ability to predict that something's going to happen.
Yeah.
And things like if we had the ability to predict when when someone was going to have a retinal detachment, we can use the current endpoint of preventing retinal detachments as a legitimate endpoint. The current problem with doing a trial in that area now is the risk of retinal detachment is relatively low. And so you have to enroll many, many patients to be able to show something. If you had a better chance in being able to predict which group of patients are likely to have retinal detachments, that makes that study doable.
Yeah, and that's why I think these kind of big collections, like Bridge to AI, like Ramin's, at least will start to contribute to our better understanding of what the indicators of risk might be, using AI tools.
Can I just ask for a clarification, Dr. Chambers, on your comment about that? So I can see that if you had a predictor of who develops retinal detachment, and those people were developing retinal detachment within a defined period of time where it was feasible to do the clinical trials, it would make sense to use that as your primary endpoint. But let's say you had a predictor that somebody was going to develop a retinal detachment, not now, not in a year from now, but in 10 years from now or 20 years from now. A therapy that prevented that retinal detachment way in the future, I would imagine, would still be of benefit. And in that case, you would likely want to use the surrogate endpoint as the new primary endpoint. Is that right, or is there something that I'm missing there?
I think the question becomes knowing that it really is predictive, short of taking it out that period of time. It is the same issue that Maureen was talking about earlier today, where we know drusen is a marker of disease going on. But I've frequently talked about it. I don't know if it's the chicken or the egg. Is it a consequence of having the disease process, and so you just see it that way? We know certainly if you get rid of it, it doesn't change the going risks. The same thing occurs here. If you think you have a predictive model for developing a retinal attachment 10 years from now, and you give it a drug product, you may have gotten rid of that particular predictive marker, but did it really change what happens 10 years from now? And I don't know of another way to do it other than to have somebody follow it for 10 years and see if it really altered the course of the disease or just altered your marker.
If I can ask, in your opinion, if there are functional sort of surrogate endpoints or surrogate endpoints that deal with retinocyclophysiology that may predict vision in a shorter time period than, say, 10 years or 20 years, are those more likely to be acceptable to FDA because we know that they're, perhaps more directly tied to the visual function?
Unlikely. The issue is it clinically relevant?
Yeah.
And if it's not going to be clinically relevant for a much longer period of time, do you really know that the natural histories with that intervention is going to be the same? And that's why we look for things that are of benefit to the patient during the clinical trials, because that's what we're required to use. We're required to use adequate and well-controlled trials to make a determination for approval. Not allowed to use anything else.
Yeah. Yeah, please.
I wanted to ask this question earlier during the biorepository, but we didn't have time. But since Wiley brought up the issue of predicting complications, it's sort of relevant now. So I'm struck by, we've heard about omics, we've heard about transcriptomics, but genetics has not been mentioned basically at all today. And even though I guess I'm not a diabetes person, I'm from an outside field, but looking on Google, I guess it's controversial, but twin studies, there clearly is a genetic role in developing diabetic retinopathy. So with all the biorepositories out there, are people doing whole genome sequencing, and are those databases being made available to really correlate clinical behavior with genetics? Seems to be obvious in so many other fields. Why are we not hearing about that in terms of diabetic retinopathy, or trying to learn from genetics more?
Does someone take a risk of answering that on the panel?
I think we are. It's called gene therapy. People are attempting to alter the local gene environment.
But gene therapy is different than genetics. I'm talking about looking at the underlying genome of how does that predict which subpopulation is more likely. It's obviously not Mendelian, it's complex genetics, but we can have genetic profiles that predict who is the best recipient for gene therapy, because gene therapy and genetics they both have the word gene in it, but they're different. Trying to understand how does a patient's genetics affect their likelihood of developing a particular phenotype.
Is there anyone else in the room with expertise who wants to try answering?
George, you want to give us a shot? The question that Dr. Zach tabled had to do with genomic analysis.
I heard the question.
Genetic analysis, yeah.
Well, the problem in general on genetics and chronic diseases has been very difficult to really doing classical genetic sequencing. So, diabetic retinopathy is a chronic disease. It takes years to form. And there have been a lot of study on genetics. There's been a lot of studies on genetics and retinopathy. In our medalist study, we have exome sequenced 1,000 patients on why they don't develop retinopathy, even though their A1C some are, been 30 years at A1C of 8 or above. So unfortunately, I think it's not going to be a simple answer, and it's going to have to take a lot more work to really understand that. For example, type 2 diabetes. genetic and type 2 diabetes. Now it's over 1,000 genes has been thought to be important. So how are we going to use that as a therapeutic approach? I think this is where the retinopathy is going to be at, is there are going to be some genes that's at higher risk, but are they going to be at a point where you could therapeutically, as a target, is a really different question.
We're missing a microphone, I think, somewhere.
I have the microphone over here. Yeah, can you guys hear me?
Go ahead.
So I just wanted to...
Stand up, please.
Yes. In the last slide of Dr. Chambers, he indicated that another endpoint could be the ability to perform an activity of daily living. So I work in the area of biomechanics, and one of the things that we do is actually measuring mobility by following people for days, sometimes for weeks. And what we're able to measure is how well people move in daily life. So we see how they slow down or whether they slow down before they get to a turn. And we do that with other medical conditions like Parkinson's. And I think that this is something that could be used in this context as well. And I just wanted to bring your attention to this.
Yeah. I don't know that we will use mobility per se, but we may use the characteristics around it. So the approval of Luxturna was done using different light level, the ability of a individual to navigate through what was called a maze, but it wasn't the issue of whether they could get through the maze or not, or how they got through the maze. It was their ability to get through the maze at lower light levels than they had been able to before treatment. So it was the fact that they could now function in a dimly lit room where they required a much brighter room in the past to go and do it. And that's the type of thing I'm talking about as far as being able to do an activity of daily living that they didn't used to be able to go and do, but now can do. And that we took as an endpoint justification of efficacy.
Can I just ask a follow-up question on that? So, I mean, I think one of the differences maybe between that example and, I don't know your name, the example of measuring mobility in the real world, right, is sort of the environment under which the test is conducted and maybe the quanta of data that you're getting, right? So if you think about, using, a tracker for, following somebody's mobility for maybe 3 months before an intervention and 3 months after, or pick a period of time, is there information there that can be still considered in the context of an adequate and well-conducted study, even though you're not necessarily directly observing, the patient, right? If you so how do you think about, I mean, it's kind of like real world type of data, but there's a lot of it as opposed to an artificial environment that's well controlled that you can, follow the patient very closely.
As long as it meets the definition of adequate and well-controlled trial, it would count. It doesn't matter whether it's in a closed setting or whether it's in a natural setting. Natural setting is frequently preferred if you can do the various things that minimize bias in the trial.
Yeah, so I mean, Laurel can give more details, but in the context of, for instance, eyes open, eyes closed, just testing of gait function and the kind of the disparity of step-to-step variability in gait speed and so forth. You can see with, eyes open versus eyes closed. With eyes closed, you get a splay of the gait attributes, whereas with eyes open, you get a narrowing, cone of gait. Those are sorts of things that, if conducted in a well-controlled trial, could become a surrogate for ambulation, mobility, activities of daily living.
So, if it occurs from the basis of the drug effect, yes, but it's not a surrogate. It's a real endpoint.
It's a real endpoint.
May I make a comment on the genomics question that Don was talking about? I think there is enough evidence in the field of diabetic retinopathy that genomics play a role. The paper by John Latchin, DCCT post-hoc analysis, he showed that the reduction of diabetic retinopathy risk by controlling sugar is explained by hemoglobin A1C control by only by 11% only. The 89%, the risk of lowering down the sugar and the benefits of the complications is by maybe regulated by other factors and probably genetic factors. DCCT has shown there is a role of familial clustering of in development of severe NPDR and PDR. Wisconsin study has shown that type 1 diabetes, only 50% of the type 1 diabetics with the longer duration of diabetes develop PDR. Only 50%. Other 50% do not develop in their lifetime. Why? There is also ethnicity variations that we see that certain populations like Hispanics and American Indians, they developed PDR more than the Caucasians. Even in our state of New Mexico, we see diabetic macular edema in American Indians is different from Hispanics' diabetic macular edema. Why? The American Indians rarely developed center involving diabetic macular edema. They rarely need anti-VEGF injections. They develop PDR for sure. Similarly, if you look at the Hispanics, they developed the worst kind of center involving DME. So the DME, there are two types, focal and center involving. They're different in different ethnic populations. So I think there is a push, there should be a push for doing more genomic studies, I'm not talking about gene therapy, to understand the disease, how the genetic factors may be modifiers, not maybe the causal variants, modifiers of the progression of diabetic retinopathy.
Yeah. Lloyd, you want to give us a little more insight on how you phenotype and characterization and kind of build the case? I mean, you led our basic and cellular mechanisms working group and the sorts of things that we're identifying there that could add to a composite profile that might help us predict risk.
Well, from the standpoint of the group we ran, it was very early, very basic mechanistic aspects behind that, for which there's been a long history and there's going to be a long future on this. Many of the things have come and found to be not so important. Other things have been important, and then there are other things that are very, very promising. All of that will take some time. I think again, to reiterate back to the concept of this, if we're going to be thinking about what other types of things to look at, is one thing that's very clear is we're able to look at many different processes, approaches, changes in the whole living system than we were able to before. And this gives us the ability to look at areas where we're going to see changes in certain things that we weren't able to before. And I think one of the mandates we have then is to, if we find these things are well associated, associate them with things that are important for visual function, or whichever, visual acuity, visual function, the critical endpoints. Because once you can do that, then, as Dr. Chambers say, you'll be able to use that as an endpoint or something to look at. So we have to discover them, do very, very good validated testing on that, and then hopefully add that to our armamentarium of things that we can use as endpoints to look into other areas as well. Both for that and particularly if we can start to get a better handle on function of various aspects. I think it's an open area where there could be lots of potential, but we have to think about the ways to do it and the things that we can find that are important to patients that are sort of outside of what we may think of just in the ophthalmologist office.
So let me shift a little bit and ask, well, okay, go ahead.
All right. This is for Dr. Chambers. Thank you for the presentation. On your slide about microperimetry and visual field testing, right, you noticed that the number of visual field locations was arbitrary, right? But can you speak to the size of the threshold change? It was 7 dB. I was just wondering if you could kind of speak to how you landed at that number.
We think 7 dB is something that everybody would agree is a meaningful change.
Yeah. Ramin, go ahead.
I have a question.
Yeah.
So we know that in diabetes, neurodegeneration and loss of capillaries are quite binded to each other. And we can quantify now capillaries quite easily. We have good machines for this. So I hear, Mr. Chamber, you say that preserving photoreceptors is acceptable because they are related to vision. So I think preserving capillaries is also something that is useful for vision. Is it possible to imagine that if an intervention, whatever is able to, in a proper manner, to show that it's preserved capillaries, so in the intervention group we lost much less capillaries than the other group, that can be considered as a sufficient endpoint, significant, and for others, if they have a point of view on the significance of this?
I think there's a very big difference between photoreceptors and capillaries. If you don't have a photoreceptor, you don't see. Period, end. That's not true of capillaries. You can lose a fair number of capillaries and not have significant change in any measurable vision that we're aware of. Does that mean there's not some level of change that is likely to be clinically significant? Yes, there probably is. But it's not the same as being a kind of one-on-one change.
Yeah.
I think that's absolutely correct. For central vision, foveal aspects, you can get quite a variation in foveal vasculature with little change in vision. Also, though, to think about capillary change throughout the rest of the retina with regard to ischemia, there is a variety of things that have been shown now that the ischemia can be associated with DRSS worsening. So you'd have to look at that in adequate detail for trials, but there's data for that. So I think we need to separate those two when we think about it. Sometimes people confuse the two.
Yeah. So I'm going to ask if, yes, I apologize, because I can't see out into the audience.
I understand.
Oh, Thomas, thanks.
Thank you. So if I understand correctly, the ellipsoid zone size was used as the primary endpoint for the MACTEL studies. That's right? Wow.
Not currently approved. I can't talk about it.
There you go.
Okay. But they had discussions with you about using that as an endpoint at some point, right?
To the extent that EZ zone measures loss of photoreceptors, it's an acceptable endpoint, yes.
Okay. So there are a lot of disorders that affect the outer retina where it's pretty straightforward. It's photoreceptors. But diabetes, the neurodegeneration is, as we understand it, is primarily in the inner retina. Ganglion cells, bipolar cells, amacrine cells. And Jenny and Paulo and Lloyd have really made the critical observation that disorganization of the inner retinal layers, or so-called drill, is highly correlated with vision in certain patients with diabetes. If there were additional data to support that as a predictor or as a response to treatment, would an anatomical index like that be of interest to you, shall we say?
So the preference in general is for functional outcomes.
Yeah.
Because
we agree, but we're still trying to find out what's going to happen.
That doesn't, obviously have taken some anatomic measures. I'm not saying that the anatomic measures will never change along there. But the type of change that you're talking about compared to a loss of photoreceptors is a very big difference.
Why do you say that? How do you know that? If you lose ganglion cells, then the photoreceptors aren't signaling.
You think there's a question that you can see without a photoreceptor?
No, but if the ganglion cells are working, it doesn't matter what's happening to the photoreceptors, they're not going to be signaling, right?
If all the ganglion cells are gone, yes.
Doesn't have to be all of them. You can lose a field defect without losing all the nerve fiber layers or all the ganglion cells.
Well, if you have a visual field defect, we'll take the visual field defect. You already have that endpoint. And I was already talking about taking a nerve fiber layer change, which is similar to what you're essentially talking about. Not
Right.
exactly.
Exactly.
But
I raised the issue of the drill because it is a more recent observation. It is more recent. It is more tied to visual acuity than any of the other endpoints, and it changes more rapidly in response to DME treatment, for example. I'm just trying to
visual acuity is no more
important.
important than visual field.
What's that?
Visual acuity is no more important than visual field.
Okay.
But the ability to accurately determine how much of an effect on seeing by some of the other areas within the retina, I don't think is as clearly defined. It may get there, but I'm not sure at present time that it is.
Okay. So, thank you. So, can we think of looking forward, because we're not resolving anything, but today is really setting the stage for the future. So it would be fantastic if we could engage you and your colleagues at the FDA in an ongoing discussion as the data from the studies that Jenny has talked about rolling out. Would that be a possibility too?
For anybody that knows me, in our division, we do not turn down meetings.
Fantastic.
Period.
There you go. Thank you.
Thank you.
Thank you for that.
Appreciate it.
I would just add, we're
Yeah,
all
thanks,
like
Jeff.
very fortunate that that's the case. If for those of you who don't like realize that that's not necessarily how like the whole apparatus elsewhere works. Now you have to be careful what questions you ask.
Indeed, fair enough. So I thought, yeah, please, Ramin.
I think we have a problem in diabetic retinopathy compared to glaucoma or GA, is that in GA and glaucoma, the disease is really spreading in a very systematic way. So it begins somewhere and then spreads, so easy to measure. So in diabetes, we have the problem that it becomes small islands of problems that appears, and we don't see with the eye, we see with the brain that process the signal. So until all these islands coalesce to each other and becomes a big area, we continue to be able to see. Not as good, and you perfectly describe. You may have difficulties to do this and that, and it very depends how your brain is trained to do this. Yeah. So for a long time, we have a problem that our patients are able to do what they want to do, not in a comfortable way. And we are not able to measure something until everything explodes because they coalesce. So I think we have to think how we can solve this problem and drill or whatever possibilities, or we can just detect these small islands and prove that these small islands have lost function, so to be able to measure something that is preventive before the real problem happens.
And at the present time, that just means longer trials.
Yes.
Which I understand is difficult. They are doable, but it is more difficult to have the longer trials. Yeah. Agreed.
So in the few minutes we have left, I don't want to not bring up an emerging problem, at least that we're all starting to recognize, which is the increased incidence of significant, even vision-threatening diabetic retinopathy in adolescents and young adults. And so we have Julie Rosenthal, who's here, and Risa, well, from Hopkins, who are particularly interested in this space. And can we have a little bit of a back and forth on involving children, adolescents, young adults in clinical trials, what our different senses in terms of evaluation and therapeutics development?
Okay, thank you. So I'm Risa Wolf from Hopkins, pediatric endocrinologist, and maybe the only one here from that area. So our kids with type 1 and type 2 diabetes really bear the burden of a lifetime of diabetes. So, while we may not see, the small vascular changes until they're teenagers, some of these, neurodegenerative changes and probably visual function are happening earlier, but to us, it feels like we're not really doing anything about it yet until the kids start to develop hemorrhages, and then even then, we're not necessarily intervening. So I think from my perspective, I'd love to see some, sooner endpoints there, because maybe we can do something to prevent what is becoming vision-threatening diabetic retinopathy in their mid-20s, which is completely unacceptable.
Yeah, I was really struck by what Ryan said this morning about, he was getting his regular eye exams, and nothing was there, and then he still developed late disease despite good control. And I think when we just look at the retina and we're not seeing anything, we're missing a big opportunity, because there, it's a neurovascular unit, and the vascular part of it is just one part of it. And I think, we're having functional changes before we see any of these structural changes. We looked at adolescents ages 7 to 17. It's very preliminary. We had about 77 kids perform an FDP test, which is similar to visual field, of course, and about 30% of them had abnormalities on that testing, and not one of them had a change when they were examined. So something is happening before we're seeing any changes. And if we could prevent, what Ryan went through by finding these things earlier, by having earlier endpoints, and then finding an intervention, I think that would be the holy grail here.
So what molecule are you proposing that they take?
Holy grail.
I say it and people laugh, but
No, you're right.
in a clinical trial, it is not necessarily the best to be in the therapeutic arm. There are plenty of trials where the placebo was the better arm to be in. The same thing goes on with trying new molecules. Sometimes you make the condition worse. You may think you're doing one particular thing, but the body compensates and you end up making the whole situation worse. It's why we require adequate and well-controlled trials before we approve particular products. I don't think you'll see the FDA stopping you from trying most different things in trying to prevent neurodegenerative disease. But you've got to be willing to follow them long enough to see if it made a difference, so that not that group, but the people in the future can benefit from that.
I might also add, there's, from the sponsor side, there's a lot of different incentives that the government has put in place to encourage pediatric research that provide very tangible benefits for sponsors of studies. But pediatric research is really hard too, right? So what's the right point in drugs development in which you would want to undertake pediatric studies, how you design them, and then how you engage with the community to actually enroll kids and their families into clinical research studies is like a whole, that's a really important discussion.
Thanks. Chris, you want to, I'll just give everyone an opportunity to have a final word.
Well, I guess I didn't have any final words prepared, but thank you all for everything that you're doing, and I hope this leads to the development of new biomarkers, the more rapid development of therapeutics, and I look forward to not experiencing those endpoints.
All right, fair enough. Thanks, Chris. Thank you all. Thank you.